Welcome back to this next video, and in this video I'm going to talk about the Punnett Square. Now, what is a Punnett Square? The Punnett Square actually is a diagram that is used to predict the genotypes of a particular cross or breeding experiment. So this is actually a square diagram that can show you that can predict the genotypes that you can expect from a particular cross or breeding experiment. And this diagram is actually a probability of an offspring having a particular genotype. So when you cross two parents, you can expect a lot of genotypes. So this particular Punnett Square actually show you the probability of an offspring having a particular genotype. And this Punnett Square is actually a tabular summary of the possible combinations of maternal alleles with paternal alleles. Because when you talk about the crosses, you are getting alleles from the maternal side, you are getting the alleles from the paternal side. So this Punnett Square is actually a tabular summary of the possible combination of these alleles. A little bit of history. This Punnett Square is called Punnett Square because it was designed by Reginald Crondall Punnett. Now, these are not three names. It is the single name of the scientist who proposed this Punnett Square, and therefore it is called the Punnett Square. Now, this Punnett Square, you can use it for the monohybrid crosses. You can use it for the dihybrid crosses, and you can use it for the multi-hybrid crosses, and to determine the possible combinations of the genotypes of the maternal alleles with the paternal alleles. Before going into the use of the Punnett Square, you should understand some of the important terms that are used when you talk about the when you predict the genotypes and the phenotypes of the offsprings. So one of the most important terms regarding the Punnett Square, that is the genotype. Now, what a genotype mean? Now, a genotype, an organism's genotype is the set of the gene that it carries. That means that the set of the gene present in a particular organism actually constitutes it a genotype. If you talk about the phenotype is actually an organism. Phenotype is all of its observable characteristics. That means you can actually see them, the physical appearance of a particular organism, and this particular phenotype is influenced both by the genotype and the environment. So when you talk about the Punnett Square, you are actually focusing on the genotypes, but this phenotype, the physical appearance of an organism, that is influenced both by the genotype and the environment. Another important term to understand is the term allele. Now, what does allele mean? This allele is actually a variant form of a gene. So if you talk about the diploid organism, for example, so when there is a gene, that particular gene can come into a wide varieties that can be available in variant forms, that can be available in different form of the varieties. And those varieties of that specific gene are called the alleles for that particular gene. I'll give you some example of the alleles later on in this video. But this allele is actually a variant form of a gene. So you're talking about a single gene and the variation that this gene can have. Another important term, that is the homozygous. What the homozygous means? Now, the homozygous means that the two copies of the same allele, and these alleles are either present in the dominant form or these are actually present in the recessive form. We will be discussing about the dominant and the recessive in a while. But when you talk about the homozygous one, both of the alleles, they are the same. Now, homo means same. That means that if you talk about a pair of the alleles and if they are identical to each other, that particular pair is known as the homozygous one. Let me give you a simple example of this. When you talk about the human beings, we are a diploid organism. A diploid means that we have two copies of every chromosome, and you all know that every chromosome carries genes. So if this is a pair of the chromosome, the homologous chromosome, we as human beings, as we are a diploid, so we get one chromosome from our mother, one chromosome from our father. the mother chromosome, say for example, it have got the capital T allele, and the father chromosome, if that have also the capital allele, that means that allele, they are the same. We say that this is homozygous status. If you talk about the small t, say for example, on the mother chromosome there is a small t, and on the father chromosome there is also a small t, that means that again this is homozygous, because both of the alleles on these homologous chromosome, they are the same. If you talk about the heterozygous one, so the heterozygous mean that one of each copy of the allele, one is dominant and one is recessive. Now again, look at this particular example. If this is a homologous pair of the chromosome, and the chromosome of the mother contain the capital T allele and the chromosome of the father contain the small t allele, is these two alleles are different from each other, therefore we use the term hetero. Hetero mean different. So when you talk about the heterozygous one, that mean that one of each type of the allele, which one is dominant and one is recessive, both of them, they are present in that particular individual. Therefore we call it is the heterozygous one, because hetero mean different. Now what a dominant allele mean? Now when you talk about a dominant allele, we say that when an allele is dominant, the characteristic it is connected to is expressed in both homozygous and the heterozygous condition. Dominant means that if that allele is present, whether that is present in the homozygous form, like in this particular form, it is going to express itself. If it is present in the heterozygous form, like this particular form, that is going to express itself. That means that that particular allele is dominant in nature. So now this capital T, now let us take this as an example of the dominant allele. So if you look at this capital T, when it is present with another capital T, it is going to express itself. But if this capital T is present with the small t, again this capital T is going to express itself. So we call this capital T as the dominant allele. Now what is a recessive allele then? So when an allele is recessive, the characteristic it is connected to is only expressed in the homozygous condition. If the say for example the small t is a recessive allele, it can only express itself when it is present with another small t. This means that this recessive allele is going to express itself only in the homozygous condition. It cannot express it in the heterozygous condition, because if you look at over here, the small t is present over here, but because of the dominant nature of this capital T, the small t is not able to express itself. That means that small t is only going to express itself when its another member is also the small t. That means in the homozygous condition, the allele which can express itself is called the recessive allele. Now as I've told you that this Punnett square can be used to predict the genotype in the monohybrid crosses, the dihybrid crosses, or the multihybrid crosses. So let us discuss let us discuss them. The first one is the monohybrid cross. Now what this monohybrid cross means? Now, if you talk about this term mono, it actually refers to single. That means in the monohybrid crosses, you are actually talking about a single trait. So mono mean that a single trait is involved. If you talk about this term hybrid, this hybrid mean that this particular trait has two different alleles. We will discuss them, like you have got a capital T allele, you have got a small t allele. Therefore, this term hybrid means that the trait you are concerned with, it has got two different alleles. Now this cross means that there are two individuals, usually male and the female, who are crossing or combining their genetic information. Therefore we call them as the monohybrid crosses. Mono means single, the hybrid means that the trait in question has got two different alleles, and the cross means that you are crossing two parents, they are combining their genetic information. Now let us take the height of the plant as an example. When Mendel he was doing his experiment, one of the trait he was focusing on, that was the height of the plant. And the height of the plant, that come in two varieties. One is the tall, and the other one, that was the dwarf. Now this tall, that was a dominant trait. That means it was expressing itself both in the homozygous and in the heterozygous condition. And the dwarf, that was a recessive trait. That means it can only express itself in the homozygous condition. Because the TAL, that is dominant, so it is accustomed to represent the dominant allele with a capital letter and the recessive allele with a small letter. So as the TAL is dominant, therefore it is represented by a capital T, and the allele for the dwarf, that is actually expressed by a small t. Now if you look at this Punnett square, I have just drawn a diagram of the Punnett square. If you look into the detail of the Punnett square, the left side of the Punnett square, like this particular side, it is actually showing you the gametes of the one parent, and the top of the Punnett square, it shows you the gametes of the other parent. So the outside of the Punnett square, that is actually showing you the gametes, that means the product of the meiosis. If you talk about the interior of the Punnett square, the interior of the Punnett square is showing you the product of the fertilization. When the gametes, they combine with each other, so the interior of the Punnett square is actually showing you the product of the fertilization. And each square in this particular Punnett square, like here we got 4 of them, each of them is actually showing 25% probability. So this box is 25%, this is 25%, this is 25%, and this is 25%. So each of the box is actually showing you the 25% probability. Now, if you talk about the monohybrid crosses, the first thing you have to keep in mind is the Mendels First Law. Now what the Mendels First Law states is that each member of a gene pair, that means that the gene that come in pair, like if you talk about this gene, they are actually in pair form. Therefore, he is talking about the pair of the genes. Each member of a gene pair, like you have got 2 members, so each of the member that have got an equal probability of being a gamete, and a gamete gets only one. What they simply mean is that the genes, they are actually present in the pair form, and during the gamete formation they separate from each other, and when they separate from each other, that can go into any of the gamete. That means they have equal probability of being in a gamete. But the important thing to keep in mind is that a gamete is going to get only one of the member of this pair. So if this is a pair having 2 members, so during the gamete formation, one gamete is going to have this capital T, the other gamete is going to have this capital T, but both of them, they cannot go into the single gamete. Same will be true for this particular pair of the this particular pair. So each of the member of this pair during the gamete formation, they are going to get separate from each other, and they will go into the separate gametes. Now if you are crossing a homozygous TAL plant, so as you can see, we are talking about homozygous. That means that both of the members of this pair, they will be same. And you are talking about a TAL plant, and as we know, we represent the TAL plant with a capital T allele. So this is homozygous. That means that both of the members of this pair, they will be capital T. If you talk about the homozygous dwarf plant, you all know that the dwarf plant expresses itself only in the homozygous condition. So the pair in this particular case will be small t and the small t. So when you are going to combine them with each other, when you are going to cross these 2 plants, what you can expect? The first thing you have to do is that you have to make the gametes. And as I've told you that during the gamete formation, these 2 members, they are going to get separate from each other. So these gametes actually will be present on the outside of the Punnett square. So one gamete will be having this capital T allele, the other gamete will be having this capital T allele. Same will be the case for this particular parent, that during the gamete formation, one of the member is going into one gamete and the other member that is going in the other gamete. So as I've told you that outside of the Punnett square, that is showing you the product of the meiosis, that means the gametes. So this side is showing you the gametes of one parent, the upper side is showing you the gametes of the other parents. In the inside of the Punnett square, as you are going for the fertilization, that means that the gametes, they are going to combine with each other. So this is one gamete from one parent, it is going to combine with the other gamete from the other parent. So this is actually again a diploid organism that will be a fertilized egg. If you talk about this one, like this, if you talk about this gamete is going to combine with this particular gamete. So you will be having fertilization. This gamete, it's going to combine with this gamete, and this particular gamete again is going to combine with this gamete. Now that means that after the combining of the gametes, you will be actually having the fertilized product. Now if you look at this one, this capital T and the small t, this is a heterozygous condition, because both of the gametes, they are now different from each other. Therefore we call that as a heterozygous condition. But as the capital T is dominant over the small t, so if you talk about the phenotype of this particular offspring, if you talk about its phenotype, its observable characteristic, that will be tall. And this heterozygous, actually this capital T and the small t, this is heterozygous, and this is actually showing you the genotype of this particular offspring. So as you can see, all of the offspring, they have got the same genotype, and this capital T small t, that is actually constituting the genotype of this offspring. But as all of them, they will be tall, so their phenotype will be tall in nature. Now if you cross the heterozygous plant with a heterozygous plant, like you have got heterozygous tall plant, this will be capital T and the small t, as the capital T is dominant over the small t, so the phenotype of this particular plant will be tall. So if you cross a heterozygous tall plant with a heterozygous tall plant, what are you going to get then? Again, you are going to make the gametes for this particular parent. So if you make the gametes, the capital T is going to get separated from the small t, and they can go into any of the gametes. So this particular side is showing you the gamete of this parent. If you talk about this one, this parent, it can actually make two types of the gametes, having the capital T, having the small t. So if you are going to cross them, that means that this gamete is going to combine with this gamete, giving you a capital T and capital T. This gamete is going to combine with this gamete, giving you a capital T and a small t. This gamete is going to combine with this gamete, giving you capital T and small t, and this gamete is going to combine with this one, giving you a small t and a small t. Now if you look at the results of this Punnett square, as I've told you, it is predicting the genotypes of the offspring. So one of the genotype that you are expecting, that is capital T and capital T, this will be the genotype. And if you talk about the phenotype of this particular offspring, that is going to be tall, and this is going to be tall, because the capital T and the capital T, they are representing the tall phenotype. If you talk about this in terms of the homozygous or the heterozygous, so this is going to be homozygous tall, because both of the alleles, they are of the same nature. If you talk about this one, like this capital T and the small t, and this capital T and the small t, this is making another genotype. This is going to be the genotype of these offsprings. But again, this is going to be heterozygous, because both of the alleles, they are different from each other. But as the capital T, that is dominant over the small t, the phenotype of this particular offspring is going to be tall in nature. If you talk about the third one, the small t and the small t, this will be the genotype of this offspring. Again, this will be homozygous in nature, because both of the alleles, they are same. But as the small t and the small t, they are representing the dwarf phenotype. So if you talk about the phenotype of this offspring, it is going to be dwarf in nature. Now, as I've told you at the start of the video, that the Punnett square is showing you the possibly probabilities, or it predicts the phenotypes. So if you talk about the genotypes in number, this capital T and this capital T, this is actually giving you 25% of the probability that 25% of the offspring they will be having this particular genotype. This capital T and small t and this capital T and small t, this is 25%, this is 25%, but as both of them they are of the same nature, that means that this particular genotype, the capital T and the small t, they will be making 50% of the offsprings. If you talk about the small t and the small t, again this is 25%. So that actually show you the percentage, the probabilities of the offspring with respect to their genotypes. Now if you are going to cross a heterozygous star plant with a homozygous dwarf plant, what you can expect then? That you are taking one of the parent is the heterozygous one, the other parent is the homozygous one. As this is a heterozygous star plant, this is star because the capital T is dominant over the small t. This is dwarf because the small t and the small t, they are in the homozygous form. Therefore the appearance of this particular plant will be the and that will be dwarf. You are going to make the gametes for this particular parent. The capital T and the small t, they are going to get separate from each other. Talk about this parent, they are going to get separated from each other. When you combine this gamete to this one, you are actually getting the capital T and the small t. When you combine this with this one, getting the small t and the small t. When you combine this with this one, you are getting the capital T and the small t. When you combine this with this one, you are actually getting the small t and the small t. Now this means that this particular genotype, this is heterozygous in nature. If that is heterozygous, the capital T that is dominant over the small t, that means that the phenotype of these particular offsprings, that will be tall, and they will be constituting, like this is 25%, this is 25%. That means that the probability of these will be 50%. If you talk about these two, the small t and the small t, this is the genotype of these offsprings. So if this is the genotype, again this is in the homozygous form, that will not be in the heterozygous form because both of the alleles, they are of the same nature. This small t and the small t, the phenotype it codes for is the dwarf. And if you look at their percentage, their probability, this is 25%, this is 25%. That means it will be having a total of 50% probability when you cross a heterozygous star plant with a homozygous dwarf plant. We will continue this discussion in the next video.